Welcome back, everyone, to non-Mendelian genetics. Today we're going to be finishing our topic of non-Mendelian genetics, but first a quick review. Last we left off, we were talking about incomplete dominance, which is right here. And incomplete dominance means that we get the heterozygote giving us a third phenotype. So for example, here we have a red Japanese 4 o'clock, and it is the dominant, but the white is not completely masked, which is why we get this pink right here. So we get the red partially masking the white, which gives us this third phenotype. So this is incomplete dominance. Now, codominance is when we get both traits showing up. And so over here we have codominance with blood types. Blood typing, we get two things. We get codominance with the with the so we get codominance with the A and the B, and we also get multiple alleles, which means that there are 3 alleles for blood type versus the normal 2. Multiple alleles can also be 4 or however many alleles there are for that. And then last but not least, what we're going to be talking about today is sex-linked traits. And so sex-linked traits means it's on the sex chromosomes, not the autosomal. So real quick, autosomal means chromosomes 1 through 22, where sex links are your 23rd pair or your X chromosomes. And so calico cats, which is what you see right here, is a sex-linked trait for fur color. It's on the orange and black fur. It's on the X chromosome, and also codominance, because if a female cat has both orange and black will show equally. So you see this orange and black fur. Now, some chromosomes in classical genetics. Walter Sutton in 1902 proposed that chromosomes were the physical carriers of Mendel's alleles, which the chromosomes is our DNA, so he's right. Problems arose, however, when regarding the following question. Why are the numbers of alleles which undergo independent assortment greater than the number of chromosomes on that organism? So this was explained with the understanding of 2 additional factors, which was sex linkage and crossing over. So sex linkage, this means that it's on the sex chromosomes, which is the 23rd pair. Remember, autosomal is 1 through 22. Sex-linked is our 23rd pair. So all chromosomes are homologous, 1 through 22. So all autosomal are homologous. But the sex chromosomes are not. And the reason for that is we get an X or a Y. Now, if an organism is XX, it's female, so all the females are XX and all the males are XY. Now, if a recessive allele, like colorblindness, exists on the X chromosome, it will not have a corresponding allele on the Y. So it is always expressed. So let's look at this Punnett square right here. We have a normal father and a carrier mother. Oh, let's put a regular pen on. So we have a carrier mother. So she does not show the trait because the trait is recessive, and a carrier father. Now, when we do this cross, we get one female who is dominant for both traits, so she's normal. One female who is normal but carries the little a. One male who is normal, does not have the trait, and one male that has the trait. So that X little a, because he does not have the corresponding allele on the Y chromosome, shows. Now, for a girl to show, she must have two X's with the little a for the girl to show. Now, here's an example with fruit flies or Drosophila. The recessive gene is for white eye color, and it is, we're going to call it XW, and it is on the X chromosome because XW. All males which receive this gene during fertilization, 50% will express this. Now, if a female receives an XW, she usually will not express this because she has that other X that will allow her to, she has that other X which will allow her to have the chance of getting another chance at not getting this. So for example, so we're saying W is for white. So one will have a W and the other one will have a plus. So if it has that plus, it's not going to show up. Where for a male, he just needs that one with the W for him to show. So here, we have a male that has it and then a female that doesn't. Here's another. Now, if you look, they use XW and then they use XW and then XW plus. XW plus means that it has the red eye color or is the wild type. So you can look at it as WT. So for human sex-linked traits, we have the following: hemophilia. This is a disorder of the blood where clotting does not occur properly. And this is just due to a faulty protein. And this occurs on the X chromosome, which means that girls have two chances to get the correct one, where guys only have one chance to get the correct protein. And so a vast majority of those affected are going to be male. The first known person to carry this disorder was Queen Victoria of England. So a lot of those who are affected in our European bloodlines are related to Queen Victoria of England. So let's look over here. So we have a carrier mother, so she's X big A, X little a for hemophilia, and a normal father, so he's X big A Y. Now we have a normal male, so he's X big A Y, and a normal female, X big A, X big A. An affected male, so he got the little a and the Y, which means he's going to show that he has hemophilia. And then an affected female. Well, if you look, the female's not colored in, so she's going to be affected in the carrier. So we have one carrier female, a normal male, a normal female, and an affected male. So here is a pedigree of the royal family. A couple of things about a pedigree. Squares are male, circles are female. If there is a line between them, it means they are married. If there is a line going through diagonally through one of them, it means death, or you can see a number underneath them. That means the age they died. If they are filled in completely, that means that they are affected with the trait that we're looking for. If they are halfway filled in, it means they carry that trait, or you can also see it as a dot in the middle. That also says that they carry that trait. If there is a line going down from a horizontal line, it means that they have a child. So the mating resulted in a child. So another couple things to note is when you look at the over here, this tells you which generation you're looking at. So 1 results in this very top one, where 7 or 8 results in the one down here. And then we number them from left to right, going 1, 2, 3, 4. So right here on 8, Charles is 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 people in row 8. Okay? So quick things on that. Now, hemophilia in the royal family. This is the royal family of England, starting with Queen Victoria, who was the first known carrier of hemophilia. So we can follow her line down, and we can see that right here, we had 2 females that were carriers and 1 male that was affected or had hemophilia because he's fully filled in, so he has the trait of hemophilia. Now we can go and look, and he died at age 31, so he was able to have kids. So let's look down. He had a normal male son, because remember, he does not pass on that little A. All he passes on to the son is the Y, because, you can't be a boy without a Y. But his daughter got that little A from him. So she's a carrier. So we look at her kids, and she had an affected son. Now, because this is the royal family, there's lots of inbreeding, at least back then. And now we're able to look and see a couple of things from here. But you can tell this is a sex-linked pedigree due to a couple of things. The first thing that you can tell is females are mostly carriers. You never see a male carrier, you see only female carriers. And all of those who are affected, or most of those who are affected, are filled in, and they're all or mostly males. So all those are affected are filled in, and if we look, all the ones that are affected in this particular pedigree are males. You can have an affected female, but it's rare because you need both recessive alleles. So this is why we can tell this is a sex-linked chromosome. It's a sex-linked, and it's on the X chromosome because females are carriers. If it was Y-linked, and we were looking at this dad, then all of his children would have it because it would be on the Y chromosome. And the girls would never have it. We wouldn't have any girl carriers. Another thing that you can see, if it is dominant, if it's a dominant trait, then every generation should have it. So drawing this quick one right here. So if we said this is a dominant trait, and the mom had it, and the dad didn't, then both daughters would have it, but they'd be carriers because they'd have the trait and it's dominant, so it would be filled in still. And then their kids would have it, or at least because they're carriers, if they married someone who didn't have it, like they can pass down maybe one. Now, if it's recessive, then, so for example, here we had 2 carrier parents. Oh, kind of looks like a face. You could see one in maybe every generation or every other generation having it, but you'd see a lot more carriers. So here we'll say that he had it and he married a woman who didn't, and then none of his kids, it didn't pass down. Now over here, her husband had it, and we can look over here and maybe one of their 4 kids had it. As a recessive trait is a little bit hard, you can see it, but it's both girl and boys are equally likely to get it, and that's where it's autosomal versus sex-linked. Sex-linked usually is males will more likely have it than females. Now, a big thing that I want everyone to remember, the more dangerous traits are on the recessive alleles. Now, this doesn't mean that there aren't lethal dominants, but recessive traits are more likely to be the lethal traits. And if we look here, like hemophilia, this is recessive. If we look at this, we can see by following it that you're more likely to get recessive traits through inbreeding. So, for example, let's follow Queen Victoria. So we're following her line. Marries someone outside of the family right here. Brings that down. The son right here has is married to this pairing right here. So he's married to this woman. They're related. Go down, look, continue to look through the family. We can do the same thing over here. This person's related. And they're married here and here. We get a mating from there. And then if we looked here, like they all, 2 of the 3 had it. The more often you inbreed, the higher the chance of the recessive trait being pulled in. This is called genetic bottlenecking, which means that you are limiting the amount of genes that can show up, which means the recessive traits are going to show up more. So right here, gene interaction, this says that many biological pathways are governed by multiple enzymes involving multiple steps. And if one of these are altered, then sometimes we don't get the correct product. And that can change how a gene is shown. And then environmental effects. This is when some genes will not be fully expressed just due to environmental factors or external factors. For example, human height may not be fully expressed if individuals experience poor nutrition. So you could have a gene that says you're going to be 6'10", but if you don't eat correctly, you could be stuck at like 5 foot. If that. Here's an example, hydrangeas, which is this lovely flower right here. In normal pH, so basic pH of 7 soil, they get this blue color, where if they are in a soil of high pH, it can change the color to pink. Right here, this is the average phenotype of America. Currently, what they did was they created this woman through a computer mixing several bases from different races, male and female, and this is what the average phenotype of America currently looks like. This was back in a Special Times issue a few years ago. So a couple of things about technology and genetic testing. So genetic testing, we can see carrier recognition. We can see if we carry certain genes or not. We can do newborn screening, which allows us to look for things like Tay-Sachs or anemia and other things. And then fetal testing. So this is amniocentesis, chronic villus sampling, ultrasounds, and fetoscopy. These are all different types of genetic testings that we can use. All right, boys and girls, well, that's the end of this genetics video. Next time we meet, we're going to be starting to talk about evolution. See you then.